guys, so what I'm gonna do here, I'm gonna finish up the lecture on the cell cycle. So if you remember, we talked about how the cells do reproduce, whether we're talking about prokaryotic cells, which have their own unique cell cycle, and eukaryotic cells, with the difference being particularly on the phase, mitotic phase, which is unique of the eukaryotic cells. Obviously, this is a process which is very tightly regulated, and the regulation we'll discuss here refers to the cell cycle in eukaryotic cells, by the way. This is very tightly regulated. Why it has to be very tightly regulated? The simple reason is that you don't really want to have certain cells to keep replicating if for some reason they are not fit to work together with the other cells in the body. For example, imagine you start having some cells that have accumulated a high degree of mutations. Do you really want them to keep growing in your body? No. So for these reasons, cell and the cell cycle, there are some mechanisms that basically check whether the cells, at every step or different steps, whether the cells are fit to progress through the cell cycle or whether they should stop replicating or not. These are regulated mostly by these proteins, or the combination of these proteins, I should say, cyclings, which you'll see they are expressed at different levels in different phases, and these enzymes called cycling-dependent kinases, which are regulated by cyclings. And these proteins, you'll see, instead their concentration is constant throughout all the phases. We'll discuss that in a second. And this is done at different steps of the cell cycle, which is what we call checkpoints. So one important regulation is done, for example, in the G1 or GAP1 synthesis phase, G1S phase checkpoint. This basically is a checkpoint which checks for the presence of DNA damage before the DNA is even replicated. So does the cells contain too many mutations, deletions, and so on. So this checkpoint checks if everything's okay in the parent cell DNA, and if everything's okay, allows the cells to move to the S phase. Now, after the S phase, there's the G2 phase. Now there is a second checkpoint before mitosis takes place. There's a second checkpoint which basically checks whether the new DNA that has been made, so the replicated DNA, it's okay or not. Do we have an even number of chromosomes, or is there any deletion that took place? So that's the second main checkpoint, which is called G2M checkpoint. And finally, there is a third important checkpoint, which is called the mitotic spindle checkpoint, where the cell basically checks whether the chromosomes are properly attached to the kinetochore microtubules. Do you remember those microtubules in the mitotic spindle, which are responsible for the separation of the sister chromatids in the two new nuclei? And so it controls that everything's properly working right there too. So as I said before, these checkpoints, they're all regulated by these two groups of proteins, one group called cyclins and the other one called cyclin-dependent kinases, or CDKs. Now notice the name cyclin-dependent kinase reflects the fact that these enzymes, which act through phosphorylation, like the enzyme that are involved in signal transductions, they are dependent on some cycling, on the binding of some cycling. So the function of the cyclings is actually to turn on certain kinases, specific kinases in every step. So now the concentration of the cyclin-dependent kinases is constant, so it's always there. But if they're not bound by a cyclin, they're inactive. If they're bound, they actually get turned on. And so for this reason, the concentration of the cyclins is actually varies throughout the cell cycle. You see here what we mean for four main cyclins. Your books focus only on these cyclins, these three cyclins here, so the G1S, the S cyclin, and the M cyclin. So these are the only three that we'll discuss. As you can see, the G1S cyclin gets expressed at the end of the G1 phase. So the gene is actually turned on. So this cyclin is produced. It peaks in the transition, and then it goes down. The S cyclin instead its expression starts at the end of G1 phase, goes on through the S phase, it peaks in G2 phase, and then gets turned off. And the M cycling, finally, the expression gets turned on through the S phase, it peaks at the end, at the beginning of the mitotic phase, and then it gets turned off. So how do they work? You can see it better in the next slide, which is this one right here. Here again, we're focusing on the G1 S cycling, the S cycling, and the M cycling. As we say, they all work by binding to specific cycling-dependent kinases. And by doing that, they let the cell move from one phase to the other one. So look at the first cycling here, G1 S cycling. If you can see, remember, its expression starts in the G1 phase. This expression is turned on only if the DNA of the parent cell is fine. If it doesn't have too many mutations, nothing that is compromising the cell function. So it's turned on, and by doing that, now you have more of this cycling here, this protein, and therefore the corresponding cycling dependent kinase, which is the CDK2, binds to the cycling and gets turned on. By doing that, this allows the regulation of many other proteins inside the cells, which allows for the transition from the G1 phase to the S phase, and therefore passing the G1 as checkpoint. Once it is done, the expression of the G1 S cycling is turned off, and therefore CDK2 doesn't do the function which does combine with this cycling. At the same time, if you see here, at the end of the G1 phase, during the S phase, the S cycling then gets turned on. This S cycling actually regulates the same cycling dependent kinase that the G1 S cycling regulates, but obviously with different function, with different targets. And so when it does that, so again, its expression is turned on if the DNA is properly synthesized here in the S phase. So gets turned on and keeps going if everything's okay. And by doing that, it helps actually the CDK2 protein to be turned on and allow the cells to actually move to the M phase. So you get the G2 checkpoint done. Finally, now there is the last cycling, so then the expression of the S cycling gets turned off. The cell can move on to mitosis. Once the cell is getting ready to move into mitosis, now there's a third important cycling, which is the M cycling, that gets turned on. And by doing that, the CDK1 protein helps the cell moving through mitosis, allows basically mitosis to take place and proceed with the separation of the chromosomes. And this happens only if the chromosomes are properly attached to the kinetochore microtubules. Otherwise, this step, the mitotic spindle checkpoint, will not take place because the M cycling would not be expressed at this high level. Okay, so that's a simple version of explaining how these checkpoints are all done in these cells, but the final result is that if everything is okay, no excessive mutation, this cycling allows through the CDK2 to go through this G1S checkpoint. If the DNA is properly replicated, the S cycling will allow through the CDK2 to go through the G2M checkpoint here, and then the M cycling here, if the chromosomes are properly attached, will allow the cell to go through this mitotic spindle checkpoint. All right. Now, what is actually influencing all of this? And so there is, if you want, you can group the events that can influence whether a cell will go through the cell cycle or not, or it's going to be stopped, dividing them in between internal events. Internal events are things that happen inside the cells. For example, if a cell accumulates lots of mutations, so where would it stop? In the G1 S checkpoint, right? As we mentioned before. So that's an example of internal events. But there are also some external events, like for example, there might be some signaling coming from far away from your pituitary gland or whatever, some other hormone expressing tissue or organ, and which would just triggers, you remember, a signal, an internal signal, which would then stop the cells at a certain point in the cell cycle. And another example is the contact inhibition, which is the one I was talking to you about with hepatic cells, right? Hepatic cells would divide as long as there is room, but once the entire tissue is filled, just by contact inhibition, they will stop dividing. So that's another example of an external event. But no matter what is the event, they all act through the expression of cyclins, and therefore the enzymatic activity of the complex between cyclin and a specific cyclin-dependent kinase. Finally, sometimes cells, though, they cannot really divide indefinitely. Even if everything is okay, sometimes cells are actually forced to stop dividing. And so cells can either be stopped dividing but still stay alive and be kept permanently in what we define, you remember, the G0 or growth arrest step. So the cell will get off the cell cycle, get into G0, but will still stay alive and can still influence the cell around them. When does this happen? It happens sometimes, usually, when cells that might have already accumulated quite a bit of DNA damage. They might lead to them become cancers, or when simply their telomeres are getting too short. So these cells, they cannot keep producing other cells to which at this point would have a too short telomeres here, and later on could basically lose important genes as well. So in this case, though, the cells, they still stay alive. They can still somehow influence the cell around them. They're not really killed. In some other case, though, if there is a lot of mutation taking place, the cells can actually be forced, instead of being forced into senescence, they can actually be forced to die through an inner program, which is a programmed cell death, which is called apoptosis. This is a very ancient pathway which is present in all eukaryotic cells, and basically what it does is that it's based again on these cyclins and cyclin-dependent kinases, and when it senses a lot of mutations there, what it does, it basically activates, not only stops the cell progression through the cell cycle, but actually activates this program, which consists of a series of events which is usually executed by the cascade of caspases. Caspases are a series of enzymes that cuts or cleaves, that would be the most appropriate term, that cleave each other, they cut each other one after the other in a cascade series of events. And by doing that, they basically lead to DNA degradation and the loss of mitochondrial function in these cells, and therefore these cells basically would stop working and they will kill themselves, obviously by losing their DNA and not having their mitochondria able to produce any more energy. They would just die. Okay, and this is something that happens. It's very necessary. It's a very important mechanism, this one, because first of all, it helps preventing cancer. Imagine if cells have a lot of mutations, that they have to die. And second thing, also, in some cases, they're also induced to die even if they don't have too many mutations. But for example, if you have too many cells inside an organ, you don't want these cells to keep replicating, and so you want some cells to be produced and some cells to die at the same time. So at a certain point, you reach a stable number of cells. All right, so this was all I wanted to discuss. Again, we talked about the cell cycle in prokaryotic organisms, and then we moved to eukaryotic organism. We focused particularly on mitosis, and then finally I discussed here in the last few slides how the cell cycle has to be regulated and how it is regulated, okay, in a very tight manner through the checkpoints.